Hi guys, this is the second video in this unit that's about energy and how cells use it and how they convert it into different forms. And so I'm going to talk a little bit more about energy, kind of building off last time. A lot of it will be review, especially if you are a physics student or if you've taken physics before. And then I'm going to do just sort of a very brief intro into the processes of cellular respiration, kind of where we're going in this video. It'll be pretty brief. First of all, if you look at this little slide here, it just kind of talks about the different or some of the different forms of energy that exist, which I'm sure is very familiar to you. So I'm having all sorts of screen problems. There we go. Energy is just the capacity to cause change, and there are a lot of different ways that that can happen on our Earth and in our universe. So the first one is kinetic. Kinetic energy is the energy of motion. Then you also have thermal energy, that's heat. I'll put this. Heat is thermal energy. It's related to the movement of atoms or molecules. And then cold is just the absence of heat, the absence of that energy. Potential energy is energy that exists because of an object's location or because of its structure. So we talk about potential energy in biology. It's often related to the structure of the molecule, such as ATP, which we'll talk about very soon. And then chemical energy is potential energy specifically related to a chemical reaction. And then free energy, which I talked about related to enzymes, which is the energy that is available to do work. So if you are a physics student, you're very familiar with that work, what that means. Okay, well, moving on. All right. So here's just a fun little diagram that's showing different energy moving around in this lake. So you have the diver on the top. He has a lot of potential energy because of his location. So being above the water gives him potential energy. As the person is diving into the water, they're converting their potential energy into kinetic energy. So the kinetic potential energy is decreasing and the kinetic energy is increasing. The diver in the water has low potential energy because of his location. So the fact that he's in the water gives him lower potential energy. And then as the person climbs up the stairs, they're converting their kinetic energy movement, climbing up the ladder, into potential energy. So this is just a diagram that shows this sort of cycle of potential and kinetic energy being recycled and reused all the time, which brings me to the laws of thermodynamics. So two laws of thermodynamics. The first one is that energy is never created or destroyed, so it's just being converted from one form to another, as we saw in that diver picture from above. Of course, it can be converted into other forms besides potential and kinetic. Those are just the ones that happen to be in the diagram. And then the second law is that every energy transfer, so anytime the energy changes into a different form, it increases the entropy or like the randomness of the universe, which is obviously a bad thing, kind of. I mean, you're like descending into disorder and chaos. But there are biological processes that sort of maintain the order and offset the move towards entropy. But entropy is just randomness and disorder that's caused by the energy transfer. Okay, so now let's apply this to biology, which is what we actually came here for. So here we have a bear that I'm going to use to kind of demonstrate these laws of thermodynamics. So the first one is that the bear is consuming food. This is the same thing that happens when you consume food. So the chemical energy in the food will be eventually converted into like kinetic energy. It will be converted into potential energy in the form of ATP through the process of cellular respiration. So what's happening here is chemical energy is horrible. Not chemical energy, which is food, is used to produce usable forms of energy through cell respiration. And that's what's happening here. Now, in B, the second law of thermodynamics is demonstrated because most of the food that you eat actually is lost to the universe as heat. And that is sort of the entropy situation happening. Here we go. Also being released, of course, is carbon dioxide, which is being exhaled, the same as you, and water is a product of cellular respiration. So I'll just make a note over here that says most of the energy from your food, which was chemical, will be lost to environment as heat. And then the carbon will be used to actually like build up your body, your DNA, your more proteins, all that stuff is carbon based, like we learned in the biochem unit. So the carbon atoms that came from your food will eventually be used to build up like more of you to allow you to grow, which is why you have to eat in order to grow. So there you go. Okay, so let's talk more about metabolism. So, like I said last time, your metabolism is all of the chemical reactions happening in your body, which includes both anabolic and catabolic reactions. So, catabolic reactions are the breakdown pathways. Breakdown. I'll just put over here that anabolic are the buildup. So, if you're going to build like new DNA, so you could do mitosis, that would be anabolic. If you're digesting food, that's catabolic. Um, And like I said, also in the enzyme video, these things kind of go together. So you have catabolic pathways so that you can have anabolic pathways, so that you can use the parts of what you broke down in order to build up um, something new, like your muscle or new cells. Like I was saying before, some molecules have potential energy just because of the way that they're arranged. And when the bonds are broken in between those different atoms, this releases energy. So typically, catabolic pathways are going to release energy, and they're often going to be exergonic, which means that energy is released, and a lot of times that's also in the form of heat. So an example of that is cellular respiration, where food is being broken down, glucose is being broken down, energy is being released from that. And yeah, the opposite of that would be an endergonic reaction, which absorbs energy, and that's photosynthesis, because what happens is the plant takes the light energy from the sun and absorbs it, stores it in the form of sugar. So these processes together, photosynthesis and cellular respiration, are cyclical. And we animals and other organisms that do cellular respiration depend on photosynthesis in order to get the sugar that we need to produce our energy. So they are a cycle, and we are all depending on each other. If you want to go like circle of life, that is the case. That's what's happening. So let me just make a note about this over here. So I'll say that cell respiration is exergonic because breakdown of, just move this, glucose releases energy, say free energy. And then photosynthesis, so go over here, is endergonic because storage of light energy in the form of glucose. And the breakdown of glucose releasing free energy is then stored as ATP or in ATP. And then what's happening is this is constantly cycling so that these equations sort of balance each other out and offset one another over and over and over. So there you go. Okay, going back over here. So if we're gonna like take this up a notch to different types of organisms, there are two types. There are autotrophs and heterotrophs. Autotrophs are going to be things that are able to capture and store free energy or energy from the sunlight or a chemical. And heterotrophs are going to be things that are going to use catabolic pathways to break down molecules, excuse me, that they consumed. So autotrophs are going to be things like plants, which are going to use photosynthesis, energy from the sunlight. And normally the things that are doing chemosynthesis are bacteria, like extreme bacteria that are in the kingdom Archaea that are using like methane or other like small chemicals to produce energy. They're usually living like near hydrothermal vents or in like strange areas where nothing else really can live, but they can actually use chemicals and small molecules to produce energy instead of using the sun. Heterotrophs are going to be things like animals, um, humans, which we're animals, but I'll put us on here. Also, bacteria, fungi, and protists, which are things like amoebas. Um, for us, we're that first bullet. We metabolize carbs, lipids, and proteins. Most animals, that's what they do. Fungi, too. For fermentation, that's going to be things like bacteria and yeast, which we're using in the lab this week. They're going to do fermentation, which is a method of producing energy that's not as efficient as cellular respiration is, and we'll kind of dive into that next week. But they are able to produce energy without using oxygen, and that's fermentation, which leads to things like beer and bread and all that stuff, kimchi. Okay, let's do a quick overview of cellular respiration. And this is something that has a lot of vocab. And we're going to delve into each step sort of individually as we go through this process. And then we'll do photosynthesis after. But there are three main steps to cellular respiration. Overall, like I already said, the goal of cellular respiration is to get free energy out of carbohydrates. And all of the processes in cellular respiration are accomplished because of enzymes. So that's why we talked about enzymes first, because without enzymes, cellular respiration would not be able to happen, which means that you would not be able to extract the energy from your food and you would never have lived. You wouldn't have survived, and no life would have. So enzymes are a super crucial part of this. The three steps are called glycolysis, the Krebs cycle or the citric acid cycle, and the electron transport chain. So here they are. I'll highlight them: glycolysis, Krebs cycle, electron transport chain. All of this takes place within your cells. For eukaryotic cells, which is what we're really going to focus on, a lot of this takes place in the mitochondria, which is actually the reason that the mitochondria is considered like the powerhouse of the cell, which you've been saying since like third grade. It's because cellular respiration happens in the mitochondria. The first part, though, glycolysis, does not happen in the mitochondria. It happens in the cytoplasm. We'll see this in a picture in just a second, but I'm going to write it down for those of you who are like into words. We'll say this occurs in cytoplasm. And what glycolysis literally means is sugar splitting. So it takes a glucose, which is a 6-carbon sugar, chops it in half to make 2 pyruvate molecules, which pyruvate is a 3-carbon sugar, which makes sense because you chopped a 6-carbon sugar in half. So now you have 2 3-carbon sugars. And then also what happens is kind of like a step 1.5 that's not really part of glycolysis, but it's not part of Krebs cycle, where a coenzyme converts pyruvate into something called acetyl-CoA, which is, or coenzyme A is what it's called. But it's just kind of like an intermediate molecule that needs to exist before we go to step 2. So I'll just say that the products are pyruvate slash acetyl CoA. And the reactants, which I should have written first, but I didn't. Oh well. Reactants are glucose. So you don't need any oxygen at all to do glycolysis, and all living things are able to do it, including plants. I should have mentioned before that plants also do cellular respiration. They don't only do photosynthesis. This is step 1. Okay, so now step 2 happens. So now we've taken our acetyl-CoA and it is moved into the matrix of the mitochondria, which you might remember, especially if mitochondria was like your organelle that you chose for the election, but the mitochondria is divided into different sections. So since I'm talking about this here, I'm going to just draw a little mitochondria down here. That's the outer membrane. And then inside of that is the inner membrane, which looks like a squiggly line because it has these folds in it to increase surface area, which you'll see why. And then inside of that is the matrix. So this space is the matrix. This is the outer membrane. This in between the two membranes is called the intermembrane space. Between the membranes. And then, of course, the inner membrane, which is squiggly line. Oops. Inner membrane. Okay, now that we did that little detour into mitochondria, we can talk about the Krebs cycle. So in the Krebs cycle, there is a series of enzyme reactions that only produce 1 ATP molecule for each turn of the Krebs cycle. Now, for each glucose, there will be 2 ATP because the reaction happens 2 times. The Krebs cycle also will produce some really important electron carrying molecules that we'll talk about later on next week. So for step 2, I'm going to say occurs in mitochondrial matrix, and the products are 2 ATP per glucose, which is 1 ATP per cycle, but 2 cycles because there's 2 pyruvates. And then also the other like things that are produced are electron carriers, which will be important later on. The reactants were just acetyl CoA, which used to be pyruvate. Products, reactants, products, reactants. And that is step 2, that's Krebs. And then finally, step 3 is where things really start to become super crucial, and that's the electron transport chain. The electron transport chain happens in the intermembrane space and also on the inner mitochondrial membrane. So the enzymes of the electron transport chain are embedded in the inner membrane of the mitochondria. So it's using both kind of the inside of the mitochondria and the intermembrane space as a way to create a concentration gradient of hydrogen ions, and that's what actually like drives the production of ATP is this chemiosmosis where the hydrogen ions are going to flow through ATP synthase, which is a crazy cool like spinning enzyme, which we'll look at next week, but it like rotates like a turbine and the hydrogen ions turn the turbine. And that creates kinetic energy that allows ATP to be formed, like in huge amounts, huge, huge, huge. So if I'm going to summarize this is in the same on my slide. The products are ATP. I'll just say like lots. And also water. The reactants are oxygen. And it also uses the electron carriers. So I'll just say electron carriers from Krebs to allow the gradient to be formed. And we'll go into much more detail about this. So let me scroll down and show you this in pictures. For those of you who are very visual, I prefer to see it in pictures. So here we go. We're starting with glycolysis over here in the cytoplasm. Cytosol is just another word for cytoplasm. Then here's like step 1.5, which is the acetyl-CoA being converted into, the pyruvate being converted into acetyl-CoA. Here's step 2. This is the Krebs cycle. Krebs produces like some small numbers of ATP and also the electron carriers. NADH and FADH2 are the electron carriers that I was referring to. And then finally, step 3 is the electron transport chain that creates tons and tons of ATP, which is why that like lightning bolt is a little bit bigger. Just to highlight a couple of the other words that are on here that are important, I'm going to make a note that NADH and FADH2 are electron carrying molecules. When they're not carrying electrons, they are, so I'll say when inactive, they are NAD plus and DH 2 plus. Sorry, no H there. FAD. Mad and fad. So when they pick up the electrons, they also pick up hydrogen ions, and that gives them the neutral charge. Um, let's see, there was another thing I wanted to say. Oh, so the phosphorylation on here is referring to adding a phosphate to ADP to make ATP. So on the substrate level ones, it's using the substrate to add that phosphate. When it's oxidative phosphorylation, it's using oxygen, which is why oxygen is a um, reactant of the electron transport chain to add that phosphate. And that is something we'll talk a lot more about next week. So there you go. Go absorb that. You're doing a great job, and I'll see you in class. Bye.